The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.
Hello, welcome to episode 4 of the eLife podcast, which is produced by The Naked Scientists. I'm Chris Smith. This month, how photosynthesis reinvented itself 30 million years ago, and what did it take for it to do that? What disco lights are revealing about how mice socialise, and cells fooled into thinking they're dead when they're not, so they persuade other cells to commit suicide. We'll hear how that works later in the programme. First though, regeneration. When a part of the body of some organisms is lost, they can grow a replacement. But what controls this? And how does it differ from when individual cells wear out in a tissue and need replacing? Worms, it turns out, might be able to help us.
My name is Peter Radin. I'm an associate professor of biology at MIT and a member of the Whitehead Institute. The problem was how regeneration initiates. In particular, we were looking at this process in an organism called planarians. Planarians are flatworms. These are animals that are capable of regenerating anything. They can regrow new heads, new tails, or entire organisms from tiny fragments of the body. One of the questions that's central to understanding regeneration in any organism is, how does it start? You wound an animal, and then what happens to bring about all of the events of regeneration?
So how did you attack that problem?
Well, we attacked this problem by investigation of genes that are activated at wounds. And we had found dozens of genes that are turned on by injury. And through investigation of a set of these genes, we found one required for regeneration. So we did this with wound-induced genes, inhibiting them one by one using RNA interference, and found a particular gene called follistatin that, when inhibited, caused a block in regeneration.
What was the nature of the injury that you were testing out on these worms?
Well, the nature of the injury initially was complete head removal and tail removal. This leaves a middle piece that's capable of regenerating a head on one end and a tail on the other. And what happened was, quite remarkably, these animals completely failed to regrow new tissues. What made this particular defect catch our eye was the fact that despite failing to regenerate, these animals in which we had inhibited the gene follistatin were capable of living and capable of replacing aged cells during natural tissue turnover. What I mean by that is that there's a population of cells that are involved in normal adult life in planarians and involved in regeneration. This population is called the neoblast population of cells. And we know from prior work that within the neoblasts are pluripotent adult stem cells. So these pluripotent stem cells are dividing and producing new specialized or differentiated cells. For example, let's say an epidermal cell, skin cell, gets old, then a neoblast will replace it with a new young one. A gut cell gets old, neoblast will replace it, and so on. That process of tissue turnover is happening constitutively and is essential for life. And this process happened normally in the animals lacking normal follistatin function.
So the fact that you could interrupt this follistatin signaling and stop the regeneration of the gross part of the body indicates that that sort of regeneration is a different process from the tissue maintenance regeneration that's just repairing and replacing clapped out cells at a tissue level.
That's exactly right. The difference in the defect between tissue turnover, where everything looked fine, and regeneration really caught our eye. It suggested that this gene may have a really unique and prominent role in regeneration specifically.
How does the tissue discriminate between the removal of a big chunk of the body, like the head, and the loss of a few cells just due to oxidative damage? You ate something that you didn't particularly agree with.
How they distinguish between the absence of a cell that's gotten old and the absence of a set of cells from injury is still a mystery that is unsolved. But what we know is that when there is an injury that has removed a substantial amount of tissues, injuries like that trigger a sequence of events that are specific to regeneration. There's particular events with the regenerative cells called neoblasts, such as the migration of these cells to the wounds, or the continued division of these cells at wound sites, that only happens when you've actually removed something. We call the sequence of events that happen only in injuries where you've removed substantial tissue the missing tissue response. And it is these events that follistatin is required for.
Looking downstream of follistatin, what does it actually appear to be doing to the target tissue once it's produced under these dramatic injury situations?
So follistatin is induced, and what it's required for are a series of events that are specific to regeneration. So it's required for neoblasts, the regenerative cells, to specialize into replacement cells. We looked at this using eye regeneration, and neoblasts normally specialize into eye progenitors following head removal. And this did not happen following inhibition of follistatin and amputation. In addition, neoblasts display sustained proliferation at wound sites in cases in which they need to regenerate substantial tissue. This was also defective in follistatin RNAi animals. Finally, there's increase in death of cells throughout the body that occurs when substantial tissue has been removed by injury. This process of death, or apoptosis, can be readily visualized with molecular assays, and it failed to happen in the absence of follistatin.
Peter Radin from MIT. The process of photosynthesis goes back over 3 billion years. But in just the last 30 million years or so, a new and dramatically better form of photosynthesis has appeared. But it requires a considerable number of changes within plant structures to sustain it. And what Cambridge plant scientist Julian Hibbard wanted to know was in what order, if any, these changes needed to have occurred to make this so-called C4 photosynthesis feasible.
Photosynthesis is the mechanism by which almost all life on this planet is maintained, because it's the system by which CO2 is converted into sugars and carbohydrates. It provides our basic foodstuffs. Historically, it provides our fuel because a lot of fossil fuels were laid down by ancient plants. And so we're interested in how a particular flavour of photosynthesis evolved about 30 million years ago, because it's actually more efficient than the ancestral version of photosynthesis.
So tell us about the two types of photosynthesis. What are they and how do they differ?
The ancestral version of photosynthesis, we think, evolved in bacteria about 3.6 billion years ago, and that uses an enzyme which fixes CO2 into sugars, and that enzyme never had to distinguish between the CO2 molecule that it now uses and oxygen, which is now present in the atmosphere. And the reason for that was those ancient bacteria weren't in an atmosphere with oxygen, but later on some more complex bacteria evolved which started to pump oxygen out into the atmosphere. And as a consequence, the photosynthetic organisms today have to distinguish between carbon dioxide, CO2, and oxygen. At the molecular level, they're actually quite similar, and so this poor protein, which is meant to fix CO2, some of the time makes mistakes and uses oxygen instead.
And that presumably reduces the efficiency of the process.
Yeah, and the efficiency is reduced the most in the tropics and subtropics, because there's a correlation between the specificity of the protein and its ability to fix CO2. And as you get warmer, it fixes CO2 less efficiently.
So how did evolution tackle that?
So evolution has tackled it actually on many occasions. In the land plants, we think at least 60 different lineages of land plants have evolved the derived version of photosynthesis that I work on. And there are other lineages which have evolved other mechanisms to concentrate carbon dioxide around this photosynthetic protein. And we've become interested in how what is actually a really complicated trait can have evolved so many times, and the evidence at the moment is it's over 60.
When this trait evolved, what sorts of things have had to change?
In a C4 leaf, there are changes to the structure of the leaf itself, to the leaf anatomy. There are changes to structures within the cell. So cell biology changes, and there are also really distinct changes to gene expression and the spatial arrangement of photosynthesis within those cells in the leaf. So it's really quite a complicated suite of alterations which have to occur.
And your question is, in what order did those things crop up? Excuse the plant pun.
Exactly. So we are interested in whether specific traits occur early along that evolutionary path to C4 photosynthesis, or late, or actually whether they can be flexible, whether they can occur in some lineages very early on and some lineages much later on, but you still get to the final phenotype at the end.
So how did you do this?
What we did was we took about 73 different plant species, some of which used the ancestral C3 version of photosynthesis, some used this derived C4 version of photosynthesis, and some, remarkably, which have characteristics of both of those types of photosynthesis, but without the full syndrome. And we mapped onto those species which traits were visible in each, and so we characterized them as either having a set of 16 C3 traits, so that species was completely C3, completely ancestral in terms of photosynthesis, or another species might have 16 C4 traits, so it would be completely derived, and then in the intermediate species we'd have a mixture of some C3 and some C4 traits.
And I suppose if you look at the evolutionary timeline separating the ones which are fully C4 and those that are fully C3, you can ask where along that timeline some of these traits must have occurred.
Exactly. So we've estimated that some traits are very likely to occur early on in the transition from C3 to C4 photosynthesis, and conversely some always come in at the end, but there are a few which seem to be bimodal, so they have different distributions. Sometimes they come in late and sometimes they come in early, which implies that there are many flexible routes for a plant, a photosynthetic organism, to move from this ancestral version of photosynthesis to this more efficient derived version of photosynthesis.
Putting all of your findings together, what then is the bottom line?
So what we discovered was that this complex system, which has evolved multiple times, is likely to have evolved multiple times because there are many ways of getting to the same endpoint. So many roads lead to Rome, I think, is a phrase which could be used to describe our system. This very complicated version of photosynthesis seems to have got there on multiple occasions because there are many ways to reach that final goal. The other thing that we infer from our results is that the initial environmental drivers for evolution of C4 photosynthesis is unlikely to have actually been improvements in photosynthesis itself, but more likely to have been changes in the ability of the plants to use relatively low water supplies or something like that.
Julian Hibbard from Cambridge University. You're listening to the eLife podcast with me, Chris Smith. Still to come, undead cells that can persuade their counterparts to kill themselves, and counting the numbers of individual RNA molecules to quantify rates of gene expression. But first, a new way to understand complex behaviours in rats and mice. Alon Chen from Israel's Weizmann Institute has developed a computer system to log automatically how the animals interact with each other. Commenting on the work, Cambridge neuroscientist Hannah Critchlow.
This new system has taken 5 years to develop and may save hours of scientists' time on consuming data analysis. Inspired by disco lights, it works by painting the fur of individual mice with different coloured UV paints, illuminating them with a UV lamp, and then recording the movements of the nocturnal mice by night. A computer system then takes the videoed movements and generates tens of thousands of rows of data per hour of recorded footage and translates this into predictive behaviours, such as grooming, sniffing, feeding together, exploring, or simply avoiding each other. By providing a system for analysing complex social interactions between large groups of mice, this could pave the way to a greater understanding of disorders associated with altered social interaction, such as autism. Researchers can tweak the genes or environment of mice and then relatively quickly see how it affects their social behaviour. The scientists have firstly applied this technique to find out how enriched environment affects how mice interact with each other. And they found that adolescent mice that have grown up in a stimulant-rich environment, full of games and toys, were much more territorial, so they didn't share their food as much, and when they did interact, they worked in pairs rather than as a larger group. Mice growing up in more Spartan conditions were much more community-minded. So the scientists suggest that this finding in mice ties in with a common belief for humans that our modern stimulation-filled environment encourages individualistic behaviour, whilst simpler surroundings give rise to a more developed community life. I spoke to one of the authors of the study about the implications of this result, Professor Alon Chen.
So we have, in our case, a very specific study in which we just provided the mice during development, during, in a way, adolescence, more stimuli. In this case, they became, in a way, less social because each one have much more resources, in a way, that can develop its own territory, and therefore it probably needs less the surrounding compared to the basic group. Again, you can think about human societies and which have much more rich type of resources versus the other one, whether they are less or more individuals. And I think so it's easy to see the equivalent system or in human.
Alon Chen, and before him, Hannah Critchlow. And Alon told Hannah that since publishing this work, he's received requests to share the system from researchers all over the world. Under certain circumstances, including during development, large numbers of cells kill themselves. And it's clear that when one cell commits suicide, it can persuade other cells to follow suit. But how?
My name is Herman Steller. I'm a professor at the Rockefeller University in New York and also an investigator with the Howard Hughes Medical Institute. It's been typically considered that cells undergo apoptosis by themselves, that they make the decision between life and death and then carry out this programmed cell death mechanism to self-destruct. And the current study actually shows that these cells are very active in influencing each other in their life and death decision. Cells that die can stimulate other cells to die as well. This is significant because both in normal developments and in certain disease situations, large groups of cells die in a somewhat coordinated behavior, and this signal to die can be shared amongst the population and coordinated suicidal behavior is achieved, a little bit like lemmings all jumping over the cliff.
So some kind of signal originates from a cell or group of cells that have become destined to die, and other cells, what, in the near vicinity, die, or does this signal potentially work over longer distances?
Well, the surprising thing in the study was that the signal can work over considerable distances. A group of cells in one place can signal many cell diameters away to another group of cells that it's time to die. Now, I should say that initially apoptosis has been seen as something where the only thing that the dying cell signals to the environment is an eat me signal, so that the corpse is removed. Subsequently, a number of labs showed that cells can actually release signals to stimulate cell division. That seems to make sense in a situation when a cell is lost and you want to replace it. So it's a I'm dying, please replace me signal. So in this context, it was very surprising that when there is a strong stress on a tissue, that the dying cells can release signals that travel over considerable distances to stimulate more cell death.
How did you actually discover this?
Well, the postdoc in the lab studied the signaling of dying cells in the developing Drosophila wing. And she generated a strong cell death situation in one piece of the wing, and then she saw, to her surprise, that in a region quite far remote, that there was cell death at the distance.
How did you find out that it wasn't just the fact that lots of cells were dying and there was some kind of necrotic effect? If you've got lots of dead tissue, this might be inducing the death. How did you prove there had to be some signal made actively and sent from cells that are destined to undergo apoptosis, which was triggering other cells remote to die?
So we use a system where we, on the one hand, induce cell death with key signaling molecules to activate cell death program. But then we can arrest the execution of cell death with an inhibitor of the proteases that are responsible to carry out cell death. So in this system, the cell basically thinks it's going to die, but it's prevented from carrying out the real destruction. So these are what we call undead cells. They are really cells that think in a way these cells are dying, but they stay alive. And as a result of that, these cells also signal for much longer, virtually indefinitely, and allow us to look at the signaling context. So there's really no cell death and no tissue loss occurring, but still the phenomenon of inducing death at a distance is observed.
So you program these cells to think they're dead, but they remain viable and pump out whatever the signal is. You see corresponding death in other cells, in other bits of the embryo. So how did you then track down what the chemical was that was doing that signaling?
We had reasons to suspect that two candidate signals, one is the Jun kinase pathway, JNK, which has been implicated in signaling decisions in cell death, and a ligand for this pathway called IGAR, which is related to tumor necrosis factor, conserved proteins seen in both insects and in mammalian systems.
How did you then explore what those were doing?
Well, we again could take advantage of the powerful genetic methodologies in Drosophila to inactivate these pathways. So very strictly, we just take away the gene that encodes IGAR and then ask if we would stop this apoptosis-induced apoptosis. And specifically, we were able not just to remove IGAR throughout the entire tissue, but we could specifically eliminate IGAR function in the cells that would undergo apoptosis, and that would completely abrogate the induction of apoptosis at a distance, even though these cells at a distance still had normal IGAR function. So it really shows that it is IGAR required in the dying cells as a signal to travel and induce apoptosis at a distance.
But all around my body, every day, there are billions of cells committing suicide in this way. So why don't I just unleash this dramatic domino effect of apoptosis and all my tissues self-destruct? Something must be opposing the apoptosis-induced apoptosis phenomenon.
Yeah, no, that's an excellent question and something where we really don't have very complete answers. It's true that a small amount of apoptosis will actually lead to a compensatory response where the dying cell stimulates growth and regeneration. Only when there is sort of a large scale of death is apparently the signal strong enough to kill. But there must be protective mechanisms, because even in the systems we've studied, we've seen cells in between not being affected. So there is a different sensitivity that cells have, and there must be a complex regulation to protect against the unwanted apoptosis-induced apoptosis.
Herman Steller. And now to finish, how genes are read or transcribed. Looking at large populations of cultured cells, it appears that when a stimulus is applied, genes are turned on and gene products are made continuously and in proportion to the intensity of the stimulus. But if you look at individual cells, a very different pattern emerges. National Cancer Institute researcher Dan Larson.
It turns out, and this is one of the punchlines of the paper, it seems like there are lots of ways for the nucleus to turn genes off. And to turn a gene on requires lots of things to happen correctly. You need to recruit chromatin remodelers. You need to recruit what's called the basal transcription machinery. And we wanted to know how much of this one particular element was required for this gene to turn on. And the element that we were looking at in this case was a steroid. We can quantitatively change the level of this steroid, and we can look at the level of single RNAs being transcribed to see precisely when transcription starts and how many RNAs are made.
So the sort of premise here is that we know that when cells receive some kind of signal or they respond to some stimulus, that gene activity can be changed, genes can be turned on.
Correct.
But what we're not so clear on is how the cells respond in terms of how much RNA, how much gene transcription goes on in response to any given stimulus?
Yes. One of the ideas to emerge in recent years is that most RNA is not made in continuous levels. When you look in a population, you see this nice smooth increase in the amount of RNA made as a function of the amount of the upstream signal present. But when you look at a single cell level, RNA is transcribed in these short bursts of transcription, punctuated by very long pauses where no RNA is made.
If we were to zoom in on the genome at the time when that's happening, does that reflect assembly of transcription machinery or something, which is why everything's there, all the ducks are in a row, it gets transcribed, but then things stop while it resets itself? Or is there some other fundamental reason why you should get that very staccato, lots, nothing for a while, lots sort of phenomenon?
That's a good question. I would say in many cases we don't know what's happening at the molecular level. What we know is that, for example, for this gene that we study in this paper, it transcribes for about 30 minutes, and then it goes off for about 30 minutes, even though the upstream signal isn't changing at all. Now, the timescales of this are suggestive possibly of chromatin remodeling events, which may control some of the timing of this transcriptional bursting, or it may be due to the stability, as you say, of some of these factors at a promoter.
So how did you try to go forward on this and get a handle on what was going on?
So what we did is we used a system that allows us to visualize RNA in living cells. We make use of a system that Mother Nature has already used, the packaging of RNA into a bacteriophage. There's a bacteriophage capsid protein which binds a stem loop, and then that RNA contained in the stem loop is packaged into a particle. We've adapted that system, and we use these same stem loops, and we encode them into our gene of interest. And at the same time, we have capsid protein is expressed in the cell, and it's labeled with a fluorescent protein. So once these stem loops get made, the capsid protein binds, and now we can see this RNA in the microscope.
Wow, so you can just count the particles that get made, and that tells you how many RNA transcripts were coming off of that gene at any one time. What a phenomenal way of doing it. So what have you learned?
So what we've learned from that is that even when this gene is transcribing at saturation, there are long periods where no RNA is being made, again, due to this bursting phenomenon. When we increase the level of this steroid, the nuclear receptor responds to that steroid, and the gene which responds to that steroid changes the frequency of this bursting, but doesn't change the length of the burst and doesn't change the amplitude. So what you can think of this as a toggle switch that's going on and off, and steroids control the frequency of this toggle switch.
So what do you think are the implications of this? How does this inform or change what we thought before? Why is this important, the whole phenomenon?
I think one of the things that really comes out of this study is that we took a very well understood physiological response, the response of a steroid responsive gene to steroids. The biochemistry of these systems has been studied for 60 years, and the dose response is one of the most well-characterized dose responses in pharmacology. It's a billion-dollar pharmaceutical industry understanding how steroids affect gene regulation. All of these studies are predicated on this idea that cells respond uniformly, that this dose response is due to the affinity of the receptor and the ligand. What we have shown is that when you look at the single gene level, you see something completely different. So what we see is this complex frequency modulation, which would never have been guessed based on ensemble studies, and which suggests that this basic dose-response mechanism may be way more complicated than we originally thought. And it also raises the possibility that changing the dynamics of the ligand, in this case the steroid, could lead to new and improved ways to use steroids as therapeutics.
Dan Larson. Well, that is it for this month. But of course, if you'd like to follow up on any of the papers we've discussed this month, they're all freely available from eLife's website. My name is Chris Smith from The Naked Scientists, and I will be back next month with another look at what is hot in eLife. Until then, thank you for listening, and goodbye.
The eLife podcast from eLife, the open access journal for outstanding research in the life and biomedical sciences. Online at elife.elifesciences.org.